as you see the graph, the enzyme concentration is directly proportional to the rate of the reaction. What do you mean by rate of the reaction? The rate of the decrease in the concentration of substrate with time or with respect to time, or rate of production or increment in the concentration of product with respect to time. Okay. So that rate comes, the reaction rate gradually increases with gradual increase in enzyme concentration. It shows it has a direct relationship with that of the enzyme concentration. But how far it can go? As long as substrate is not a limiting factor. What do you mean by limiting factor? Where substrate is unlimited. Where substrate is limited, it's called limiting factor. Where substrate is unlimited, it is called not a limiting factor. Correct? If the substrate is limited, then after some time, however amount of enzyme you add to the system, it will not increase the rate of the reaction any further. Why? Because more hands, more work, fine. But if work is same, work is stipulated, restricted amount of work, then more work means, more hand means same amount of work will be done, because after this point, substrate concentration remain constant. It is not increased any further. That means substrate concentration is a limiting factor. So when the substrate concentration is a limiting factor, we have a graph like this. When substrate concentration is not a limiting factor, we continue this graph to the infinity. Clear? So more hands, more work, more enzyme molecules, more amount of product form, or higher the rate. Temperature, the second point, temperature. With growing temperature, usually the rate of the reaction increases. See? The rate of the reaction slowly increases with increase in temperature, starting from 10 degrees Celsius, 20, 30. When it comes highest, 37 degrees Celsius, that is our body temperature. Fine. So till that temperature, usually most of the enzymes show an gradual increase in the rate of the reaction. Why? It has got three parts. This part, this point, this area, and this area. These three portions of the graph you have to explain. This area of the graph is explained like this. With increase in temperature, the effective kinetic energy of the participating molecule increases, hence rate of the reaction increases. Clear? When it reaches maximum, what is that? It's an optimum temperature at which the enzymatic rate of reaction reaches maximum. It's called optimum temperature. Correct? Next, further increase in temperature causes thermal degeneration of the protein, leading to decrease in rate of reaction. So initially, increase in kinetic energy, increase in rate of reaction, then it reaches maximum at some point of temperature, then it rapidly falls. See, while increasing the temperature, it gradually increases. While falling, it falls rapidly. So it is not exactly bell-shaped graph. When you explain that a bell-shaped graph, so it is not exactly bell-shaped graph. Now most of the ectoenzymes and endoenzymes, they have their optimum temperature between 30 and 40 degrees Celsius. Correct? Except few. Say for example, Taq polymerase, correct? The enzymes produced by the bacteria found in the hot natural spring. So just remember the graph. Here you have to mention at least 3 examples showing their optimum pH, one of them in the acidic range, one of them in the neutral range, one of them in the alkaline or basic range. So here x-axis shows the range of pH. What is the maximum pH could be in the scale? 14, correct? So do not extend your graph beyond 14. V0 means the velocity or initial velocity. Both are partially correct and incorrect. Anyway, now this one shows pepsin, green one. The pepsin enzyme has its optimum pH around between 1 and 2. Between 1 and 2. See, 1 and 2. This one, salivary amylase, is between around 8. Slightly moved this way. So around 8 it should be here. And alkaline phosphatase between 10 and 12. Between 10 and 12. So just rectify this. So beta bhul ache, thik kore nao. You know? Pepsin between 1 and 2. Salivary amylase around 8. Around 8. Alkaline phosphatase between 10 and 12. So you can just keep a 10.8 or 11.2 like that. Optimum pH. Any other enzyme? Acid phosphatase between 4 and 5. Just draw all the graphs. Acid phosphatase between 4 and 5. So now you know 4 examples. Pepsin between 1 and 2, acid phosphatase between 4 and 5. Then alkaline phosphatase around 8. Hi, alkaline phosphatase amylase hoche around 8. 8.5, 8.6 like it comes like that. Fine. Now next is substrate concentration. Now if you see, if you increase the substrate concentration and enzyme concentration is constant, then what will happen? Slowly the rate of the reaction will increase. But after some time, since the number of enzyme molecules are fixed and not increasing any further, then whatever amount of substrate you add, they cannot increase the rate of reaction any further because number of enzymes are fixed, the number of hands are fixed. They cannot do more than their hands. So the graph becomes like this. Correct? What do you call this kind of graph? Parabola. Parabola. Elliptical, parabola, parabolic. Hyperbola means sharp. Parabolic graph. Now, this is not parabolic actually. Whatever it is, the graph is having its own unique shape. It is neither hyperbola, neither parabola, nor parabola, nothing. It has its own. It resembles somewhat with that of the bacterial growth graph. Lagging phase, leading phase. So like that. But this point is fixed, after which it does not increase any further, the rate. This point, when extrapolated on y-axis, this point is called maximum velocity or Vmax. X-axis is concentration of substrate. What is the unit of concentration of substrate? Moles, grams per liter, moles per liter, correct? So like that, moles per liter is given. So y-axis shows the maximum velocity at this point. Why? Because enzyme substrate enzyme is saturated far more substrate than it can deal with. See, they have excess substrates, these red dots, but no free enzymes. So, it cannot increase the rate of reaction any further. The number of molecules is fixed in case of enzyme. If you go on increasing enzyme any further, then again the graph will start growing. Clear? So, from this point, if you extrapolate on y-axis, you get a point called Vmax or maximum velocity. Now, this is somewhat an unique property of the enzyme and the substrate duo. If you change any one of them, then this Vmax might change. Clear? Now, I would urge you to write down this equation like this and this also. And also request you not to forget this reaction ever. There are a lot of stories behind this small equation we will come across, we will slowly we will discover. First of all, you just write, this equation is called, be careful with the spelling. He was male, he was female. Did you know that? Jante? Maintain Mohila ছিলেন? Huh? Wow, very good. কোন স্কুল? কলকাতার? রানীকুটি? মেরেছে, বস। So, Michaelis-Menten equation says V0 is equal to Vmax into substrate concentration divided by Km plus substrate concentration. है ना? Okay? মনে থাকবে? এখানে V0 কি? Initial velocity. Initial velocity কি বোঝো? What do you mean by initial velocity? দেখো, listen one thing very carefully. The first year is just for getting the definitions right. You should know what is what very, very clearly. That's what the first year is all about. Unless you know these things, later on it will be a problem. So, learn each and every term and terminology very carefully so that you are ready for the next set of construction. V0 initial velocity means what? Anyone? বলো, তুমি বলবে? কে বলবে? Anyone? What do you mean by initial velocity? Why not V? Why V0? Time is equal to 0. The time says, time is a variable. So, it can be T0, it can be T is equal to 1, T is equal to 2, T is equal to 3, T is equal to 4. But each time, each growing value of T will bring about some changes in V. Correct? Minus dSdt or plus dPdt. But that is not in a straight line. Because of what? We are not taking average, rather we are taking differential. কেন আমরা dSdt নিচ্ছি? কেন আমরা S by T নিচ্ছি না? The question is why we are not taking S by T? Why we are taking dSdt? Because with every 1 second change in t, the rate of change in V is not equal. So we can take instantaneous velocity of any point. For the sake of simplicity, we start with t is equal to 0. So what is that? T is equal to 0 means what? V0 means what? 0. Right? No time. That means your substrate is in your hand, enzyme is in a test tube. V is equal to 0. The moment you mix it, T increases 1, 2, 3, V increases. But the rate are not identical. That's why we cannot take S by T. We take ds dt. Yeah? So the initial velocity V0 stands because of the timing. Vmax is an extrapolated point on y-axis from the parallel point of the graph. Right? That's what. Or else, how else you can explain? It is a maximum velocity achieved by occupying all the active site of all the enzyme molecules with substrate. S within third bracket means concentration of substrate. Km. It is called, read out, Michaelis-Menten constant or Michaelis constant. Correct? Now this is something very interesting. What do you think? Who discovered this equation? Huh? It is not Namo Stadium in Gujarat. The person himself is putting his own name. This equation was not discovered by these fellows. They discovered something else. Say around after 50 years of their discovery, another group of scientists came. Briggs and J.B.S.-Haldane. J.B.S.-Haldane you heard of? There is a road, street in Calcutta by his name, Haldane Avenue. So these two fellows, Briggs and Haldane, they gave this equation and named, showing respect to their predecessors, that let us call it Michaelis-Menten equation. And they brought about some changes in Km. Michaelis and Menten first coined the word Ks, not Km. Briggs and Haldane changed it to Km. M showing respect to them and named it Michaelis Constant. Clear? So what was the difference between the thinking or thought or perception between these two group of scientists? Michaelis and Menten were chemists, pure chemists, and Haldane and Briggs were biochemists. There is a basic difference. What is the basic difference? In chemistry, whether the reaction takes place or not doesn't matter. Why? Because mother of any molecule is not going to die during giving birth. Father of molecule is not going to become old with time. N2 plus H2. Do they get older with time to produce NH3? That N2 and H2 have become very old, that's why they are producing less amount of ammonia. That never happened. Correct? But in human being, the babies grow faster, we grow older, and we die. Correct? This thing happens very fast, but very fast rate. You know? Very, very fast rate. This is the change they brought in the equation. What Michaelis and Menten said, Michaelis and Menten said E plus S. E is free enzyme, S is substrate, and this is enzyme substrate complex. Correct? So the constant for this reaction, okay, the constant for this reaction is, okay, the constant for this reaction is, fine. আচ্ছা আর একটু সহজ করে দিই। Michaelis and Menten said this reaction never happens. In chemistry, this reaction, this step never happens. Whatever amount of enzyme is converted to enzyme substrate complex, the whole of it comes back to the substrate again. So, Ks becomes that's what Michaelis and Menten perceived. এটি ওদের ধারণা ছিল, ঠিক আছে? তাহলে ওদের কি ধারণা ছিল? এক নম্বর, the reaction never goes in forward direction. 2 number, the reaction produces back the substrate. So the constant is K2 by K1. But after 50 years from the discovery, when Briggs-Haldane came, they told in biological system, when proper pH temperature is provided, K3 is much bigger quantity compared to K2. Right? So, the new constant became this, that is what? The K1 is production of enzyme substrate complex, rate of production of enzyme substrate complex, and these two together is the utilization of enzyme substrate complex. It can be utilized for production of product or formation of substrate back. Any reaction? So here they said that this K3 is much bigger quantity compared to K2. Instead of writing K2 any further, we can just omit because K2 is very small quantity. Say this quantity is around 10 to the 6 and K2 is around, will you consider? No? So they omitted, it became K3 by K1. So these was the changes or alteration made by কার কার ঘুম পাচ্ছে? জল খাও, ঘুম তাড়াও। ঠিক আছে, একটু সময় দিতে হবে। You must give some time to this. Now your brain is empty. During time of examination, this question will become very difficult. If you do not understand this, whole enzyme activity will be jeopardized. Now, so these are the assumptions made by Michaelis and Menten to deduce this equation. Am I clear? What is that? They never produce any product and they completely produce the substrate back. There is no question of K3. Briggs-Haldane said no, there is no question of K2. K3 is there. K3 and K1. This was a difference. And then they termed this case as, because case is now different, so it became, this was named by these two people, not Michaelis-Menten, to show respect to their ancestors. That's all. Nothing else. And termed them as Michaelis Constant. Now, Km being an entity, it must have some unit. How do you find the unit of Km? You remember how to determine Vmax? The point where it becomes parallel. If you extrapolate, you get Vmax. The half of the Vmax you can calculate by using a scale. Suppose this distance is 6 centimeter. Then half of the 6 centimeter is 3 centimeter. So put a dot at 3 centimeter. Write the value Vmax by 2. That is half the maximum velocity. Repeat. Half the maximum velocity. Half the maximum velocity. Vmax by 2. Now extrapolate on x-axis from that point. Whatever value you get is the numerical value of Km. What is the unit of Km? What is the unit of x-axis? Moles per liter. That means unit of substrate. So what does it define? Km is the amount of substrate at half the maximum velocity. Amount of substrate required for half the maximum velocity of a reaction. Km is the amount of substrate required for half the maximum velocity of a reaction. Clear? বুঝা গেল? Like Km value, if it is high or low, how does it matter to the enzymatic reaction? Okay. Oh, this one is difficult. Achha. Let us discuss this, then we'll go to that. Okay. So 3 points. শিখলাম আমরা. Number 1, Michaelis equation. Number 2, assumptions by the Michaelis-Menten, and changes or alterations by or modifications by Briggs and Haldane. Km, Vmax, unit of Km, definition of Km. Now, steady state kinetics. Is there anyone in this classroom who has ever come across this steady state kinetics? Just let me know. It will be easy for me then. Anyone who has ever heard this word, steady state kinetics? No one? क्यों नहीं? Fine. Okay. This is a unique graph. This is a y-axis, this is another y-axis, and this is x-axis. X-axis shows time. 2 y-axis, actually one y-axis. To make it simpler, to look it simpler, they have put 2 lines on 2 sides. If you write only 1 line, that is sufficient. 2 lines are not required. Okay. Now, here is a concentration of what? Left hand side is a concentration of substrate, right hand side is a concentration of product. So, initially at T is equal to 0, what happens? See this reaction, E plus S, ES plus E plus P. What is highest? Free enzyme. See the free enzyme is the highest concentration. What is lowest? ES, lowest. What is again in the highest concentration? substrate, nothing is utilized yet. What is again the lowest concentration product? Nothing is formed so far. So, highest concentration of substrate, highest concentration of free enzyme, lowest concentration of enzyme-substrate complex or transition state, and lowest concentration of product we start with in this graph. With growing time, passing time, the substrate concentration decreases and after some time it becomes 0. With passing time, product concentration increases, after some time it reaches maximum. Correct? The free enzyme concentration decreases but does not become 0. Why? Because no reaction in this world is completely or 100% efficient, so it cannot become 0. It will have little higher value than this 0. The ES complex again, because of the same reason, cannot reach maximum. It will reach somewhat high value and maintain between these 2 points, this point and this point. You see what is changes? Between these 2 points, see, ES complex concentration remains constant, free enzyme concentration remains constant. So, if you look at any point of time between this point and this point, what you will feel? No change in the concentration of ES, no change in the concentration of free enzyme. That means reaction is not taking place. But again, at the same point, if you see the substrate and product, one will be decreasing, one will be increasing. But at any point, it will be stand still. See this point? Substrate and product are same. ES same, no change. That means DES dt 0. DE dt 0. No change means DS dt 0. Understood? That means DES dt 0. What is the steady state means? This is what I meant to say. No change in the rate of ES or E. That means what? The reaction is not taking place? No, reaction is taking place, but at steady state. So when DES dt, DE dt 0, but still reaction is taking place, is called steady state kinetics. Between this point and this point, the reaction shows steady state kinetics. So here, if we want to estimate substrate or product formation, then there are 3 areas, which area we will choose in the graph? There are 3 areas. First area of the graph is here to here. Second area of graph is here to here. Third area of graph is here to here. Which area would you choose to find out substrate and product concentration? The graph shows minimal mistakes or aberration when it is straight line. Where do you find straight line? During steady state kinetics. So between these 2 points, we must calculate the reaction rate, not before or after this point, correct? But still, there is a problem. Now, this one you write. Double reciprocal plot. Michaelis-Menten equation, little different form. This one. Just memorize it. You need not to know the deduction and everything, anything, nothing. Don't know. Just memorize. 1 by V0 is equal to Km by Vmax into S plus 1 by Vmax. কি করে আসলো ওখানে লেখা আছে দেখো, just revert it and then just separate it. And if you simplify, you get this. This particular plot system is called double reciprocal plot, or named after the inventor Lineweaver-Burk Plot. Lineweaver একজনের নাম, Burk একজনের নাম। Lineweaver-Burk Plot or equation, ঠিক আছে। এছাড়াও কিন্তু আছে, there are many other forms of representation of the graph, but we will study only this apart from Michaelis-Menten. Why? Because we are small kids, छोटे बच्चे हैं, है ना। Hello? Then what is the difference between these two forms? This form and this form? Tell me. Who was good in mathematics? তুই বল, তুই, হ্যাঁ দাঁড়া। এই ফর্মটার এই ফর্মটার ডিফারেন্স কি বল? হ্যাঁ, একদম ওপরেরটা, এইটা আর এইটা। ভেরি গুড। সো ইটস এ স্ট্রেট লাইন, আর উপরেরটা প্যারাবোলিক। বাহ, ভেরি গুড, বিউটিফুল, ব্রিলিয়ান্ট। শোনা ছিল, বসো। So we get a graph like this, straight line. So it is, don't write. You have to write, don't worry. Wait for the time. So this is graph like this. When we do like this, it becomes straight line. Now it is very easy to calculate whatever value you want. You just put there, and from x-axis, y-axis, you get the value. Had it been like a parabolic, means you don't know which point to be taken. It's very difficult. Here there are two enzymes we have mentioned. One is hexokinase, another is glucokinase. What is hexokinase? Which converts glucose or fructose to glucose or fructose 6-phosphate. What is glucokinase? Which converts glucose, only glucose, to glucose 6-phosphate. Glucokinase is nothing but an isoform or isomer or isoenzyme of hexokinase. More precisely, type 4 isoform of hexokinase is nothing but glucokinase. Why do you find the location difference? Hexokinase is found in all tissues, glucokinase is found in only liver. Only liver. Why? There must be some reason. Which organ stores carbohydrate? Liver. When do you store carbohydrate? When the carbohydrate amount is plenty, or when the carbohydrate amount is less? Plenty. So if the glucokinase, the enzyme which utilizes glucose in liver, have very low Km, what will happen? If it was opposite, had it been just opposite, if hexokinase would have replaced glucokinase, then what would have happened in liver? Less K means high affinity. So that means what? That means small amount of glucose concentration will trigger the conversion of glucose to glycogen in liver. So when you are starving for say around 8 to 10 hours and you have taken one small biscuit, immediately that glucose produced from the biscuit will go to glycogen storage, not providing enough glucose to your other cells who are starving. To avoid such things, nature has provided, or whoever God you believe, that God has made us like that. What? The liver has an enzyme which has higher came, so that it requires very high concentration of glucose. When plenty of glucose is present in the blood, when we are having a full meal, enough food to fill our stomach, the blood is full of glucose for a prolonged period, say around 2.5 hours, then it will go on storing the carbohydrate in the form of glycogen, not before that. Correct? Whereas the hexokinase is distributed into all the cells, because whenever the glucose level falls, the slight increase amount in the glucose in the blood will trigger hexokinase to take up the glucose by the cell, say RBC, brain cells, other cells of the intestine, nerve cells, they can survive. If it happens the other way, the liver will survive, we will die. What do you want? That your liver will survive and you will die. Rather, let the liver die, we will survive. How liver dies and we survive? We enjoy when liver dies. Have you done it ever? Blatant lie. When liver dies, we enjoy. We dance. Types of inhibition. So probably the last one, last factor. Presence of any inhibitor should and must. Presence of any inhibitor should and must decrease the rate of reaction. Otherwise, it is not inhibitor anymore. Change the name. So, inhibition can be of different types. Now, you can ask me, sir, for competitive, you said uncompetitive, non-competitive, but for irreversible, where is the reversible one? Opposite thing. There are many inhibition which are reversible. Yes, there are reversible. Reversible can be competitive, reversible can be non-competitive, reversible can be uncompetitive. But which are not reversible, they are only irreversible. There we do not think about whether the irreversible inhibition is due to competitive inhibition or uncompetitive or non-competitive. Try to understand this one. Now there are different types of inhibition you come across. Now, you tell me any other inhibition, type of inhibition you know. Raise your hand and tell me. Feel free to ask. Interact. Follow. না এই যে কম্পিটিটিভ ইনহিবিশন বললাম, এরকম অন্য কোনো ইনহিবিশন নাম শুনেছিস? এই চারটে বাদ দিয়ে শুনিস নি? ফিডব্যাক ইনহিবিশন নাম শুনিস নি? ফিডব্যাক, হ্যাঁ, তাহলে আমি তো লিখিনি এখানে, তুই কেন জিজ্ঞেস করি না কেন স্যার ফিডব্যাক লিখলেন না কেন? Why nobody asked? Why I have not written feedback inhibition? Why nobody has asked? Why I have written suicide inhibition? Product inhibition, direct inhibition, indirect inhibition. There are so many words in inhibition. But I have written only these four. Why? Covalent inhibition, allostatic inhibition. You can ask. Nobody asked. Why? What is the reason? Correct. Here we are pondering over the mechanism, how it actually interacts with the enzyme. Effect can be different type. If it is at the level of initial level, it's product inhibition. If the inhibitor is from the ground level, when it's 2, 3 or 5, 6 steps away, then it is called feedback inhibition. Correct? So that is all how it is placed in the pathway, where it is placed in the pathway, but not the mechanism. This is the mechanism by which it acts. How inhibitors actually interact with the enzyme's active site. Based on that, it can be reversible. That means it can bind and it can come back. It can be reversible. Once bound, bound forever. It does not require any of these. It requires something else. What is that? Formation of covalent bond. The once it is bound with covalent bond, we cannot separate it. Clear? Now, sit down. To answer this question, you have to first give the definition, then a general reaction like this mechanism, and then one example, actual example from enzyme. These 3 points you have to remember to answer this question. Everything is given in many books, all many books. I will just tell you how to write. What to write depends on you, how much you can remember, or you can quickly copy. It's up to you. So either you become expert by remembering, or you expert by making machineries for copying. We have 9 months of time. So this is the mechanism or flowchart for competitive inhibition, where the inhibitor has structural similarity with that of the substrate interface to the active site of the enzyme. পুরো সাবস্ট্রেটের মধ্যে দেখতে একরকম নয়, সাবস্ট্রেটের যে ইন্টারফেসটা, এই যে জল খাও, তোমায় বলছি ড্রিঙ্ক ওয়াটার, it helps, है ना? So it is not throughout the body, it is not look-alike with the substrate. Only the interface part is similar to substrate, where it interacts with the enzyme. So either substrate it forms with enzyme E and P, or inhibitor, either or, because it has only one chair, one seat. Either this duster can sit, inhibitor, or I can sit. So when number of duster will increase, then the competition between me and duster will increase. When it is 1:1, that means what? 50% chance. So Km value will increase by 50%. It will be double. If there are 2 molecules, 2 dusters, and me alone, then Km value will be 3 times, because 3 times me required to dislodge inhibitor to sit in the active site. Understood? This is a simple thing. So the Km value will increase, but will it change the rate of reaction? Because enzyme is same, substrate is same. See, draw it. This one, red color, na? Increasing inhibitor means it will go like this. This is without inhibition. This is, take anyone, this with inhibition. The red line is without inhibition. Any blue line is with inhibition. So as the amount of concentration of inhibitor increases, then what happens? See, the Vmax remains same, y-axis interception point, because enzyme's activity is not changing. Only the availability of the active site is changing. So Km value will increase, but Vmax will remain same. Enzyme is still young enough to change the substrate at the same rate. But substrate is coming at low rate, so product will form low rate. But here this will increase, this will move towards the origin. If this point move towards the origin, see, this is what? Negative inverse Km. That means what? Km value will increase or decrease? Increase, because it is negative inverse of Km. Correct? So draw this graph after this part.